Okay, now I'm all set. Okay. I'll record my voice. Now we're ready. Now let's move this. Okay, so we're going to try and review the muscle types and how they're controlled. Yes. I didn't cover them. I cut them. So send me an email on the study guide to cut that out. It was Kallman syndrome and androgen insensitivity. I simply decided not to cover it. No. But do send me an email reminder, and on the study guide, I'll cross those out.
And for the lecture quiz too, so chapter 4, part 1, the past slide, like 40.
Yes.
And then it's chapter 4, part 3 also.
Well, chapter 4, well, since you're asking that, let's, okay. Do this. So since you're asking, get back to move this stupid thing out of the way. Okay, syllabus. Okay, so lecture quiz, which is coming up. 3, exam. Where in the world do I have the quiz? Right after spring break. Okay, okay. Yep, so chapter 4, part 1, everything after acetylcholine, it's right there, okay? Now, is chapter 4, part 3 going to be on it? Yes, but this, let me show you. So let me go to chapter 4, part 3, peripheral nervous system, completed notes. All you have to know is what has to do with, let me let it catch up. Okay. Sympathetic responses. So the right-hand side of this, and then of course the flow diagram, and there's the key, so just study this part right here. That's for the quiz, right? And me back out of that. And that's it. So go back to the notes. And that's all you need to know. So I'm not going to ask on the quiz anything other than fight, flight responses. Of course, you got to know the drugs because that's part of fight, flight receptors. But in that PowerPoint, you can go over the red part, fight, flight stuff, the drugs that are agonists or blockers to adrenergic receptors. I'm not asking the cranial nerves. I'm not asking anything past that. Just the fight, flight for that chapter. And then, of course, chapter 4, part 1, everything after acetylcholine signaling. Yep. Does that clarify it? Yeah. Okay. All right. So I don't want to turn off the Zoom, so let's just minimize that. And I guess go back to this. Okay. So this is going to be review the different muscle types. So of course, this chapter is going to be skeletal muscle. We're going to focus on that. But then we know this involuntary muscle, cardiac muscle, smooth muscle. Okay. We know it under rest and digest. We know it under fight, flight. So, oh, my little advanced thing isn't working. So we'll do this. So this is good review. Where'd my pen go? There it is. So let's do this. So skeletal muscle, hint, quiz and lab. What is the neurotransmitter that controls your skeletal muscles? Oh, okay. What was it again? So it's somatic motor neurons release this neurotransmitter into the synapse of skeletal muscle. Which neurotransmitter is it? Acetylcholine. Okay. And what's the receptor for acetylcholine on skeletal muscles? Since it's for acetylcholine, you know it ends in, oops, sorry about that, cholinergic, but what is in front of cholinergic? Nicotinic. I told you I would hammer that into your heads until it becomes automatic. Okay, now what I've added in this chapter, which we covered before, chapter 4, part 1, glycine can inhibit skeletal muscle activity from the spinal cord, and GABA, it was on a lab quiz bonus question, quiets the brain and paralyzes skeletal muscles during sleep so you don't get up and walk away. Okay, new thing, skeletal muscles have the fastest contraction speed. They're also the strongest, but they are prone to fatigue. And we've covered that before because we've talked about when your skeletal muscles are pushed, they go to the anaerobic metabolism and they produce lactic acid and a little bit of ATP. Okay, review of cardiac muscle. We have no control over it. It's autonomic. It can be controlled under the parasympathetic nervous system, rest and digest. What's the neurotransmitter for that? Had an exam on it before. Acetylcholine. And its receptor is muscarinic because you must. See, you're going to be set for nursing pharmacology if you remember that. Now, if it's sympathetic stimulation, upcoming quiz, that neurotransmitter is epinephrine, known as adrenaline. Oops. Okay, and its receptor is going to end in adrenergic. But what is the receptor found on the heart? Beta 1, one heart. Okay, and that will speed things up. We're going to talk about in cardiovascular system how it's autorhythmic, but don't worry about that. Intermediate speed of contraction, fatigue resistant. This muscle goes 24/7, 365. And the day you die is when it stops, right? So it doesn't fatigue. Now, smooth muscle, if it's rest and digest, but some things slow down and some things speed up. So I have to be careful. But acetylcholine with muscarinic cholinergic receptors can speed some things up, like your GI tract. All right. Epinephrine and alpha adrenergic receptors are going to slow down your GI tract activity during fight and flight. Beta 2, it's not shown here. Okay. Where do you find the beta 2 adrenergic receptors in the body? It's smooth muscle, your bronchioles. Okay. And that will cause them to open up wide. Now, what about the beta 2 best to get away, where you'll also find receptors for this epinephrine in the body that's best to get away? Arteries going to skeletal muscles. Okay, so you'll want to know that for the quiz that we have in lecture. Okay, so epinephrine binding to beta 2 adrenergic receptors causes bronchodilation and artery to skeletal muscles. I'm going to put something on top of artery. Let's put skeletal M. Vasodilation of arteries to your skeletal muscles, because that's what makes it best to get away. Epinephrine binding to alpha adrenergic receptors. Oops, always wants to do that to me. Go away. There we go. Causes decreased GI activity. And also vasoconstriction, and that's under GI activity. Vasoconstriction, arteries to GI, running out of space, so we'll just do that. So that's all stuff you're going to want to know for the next lecture quiz. But I think that you're probably getting it now. All right, we spent enough time on it. Okay. So what we're going to do from here, we're going to go over muscle organization. I'm going to try and get to, watching my time, how muscles contract. So again, I might jump around this lecture and come back to stuff I skipped today on Wednesday, but I want to go over stuff with you. So skeletal muscles, you know this from anatomy real quick. So a muscle organ is what you learn. This could be the biceps brachii muscle for all I know, and the tendon that attaches it to the bone. Now inside a whole muscle organ, this big thing right here, it's made up of subunits. Okay, this here, this here, this here is a fascicle. All right, so muscle organ, and then a subdivision within it is the fascicle. You ever seen those nesting dolls? Where you open up the doll and inside is a smaller doll and you open up that doll and there's this, that's how muscles are arranged, kind of like peeling the layers of an onion. Now within one fascicle, it's made up of many small units called a muscle fiber. That is a muscle cell. A muscle fiber is a muscle cell. And so you probably did not go to that level in anatomy. You probably stayed at the muscle organ, maybe learned the subdivisions that I just talked about. But I'm going to be talking at the muscle cell level or fiber level for how muscles contract. Now, is there anyone who took anatomy where they actually went over the sliding filament theory of contraction? Fine, because that's physiology. So that's what we're going to go over. If you pull out a single muscle fiber, one of these things, inside a fiber are thousands back to back of these things called a sarcomere. And that's the level we're going to for muscle contraction. All right. So inside a muscle fiber is a myofibril. And each little subdivision here from this end to this end is a sarcomere. And so that's what we're going to be talking at. So vocab for what we just covered, which I assume is review of anatomy. From large to smallest, the muscle organ is the muscle you learned in anatomy. That is made up of subunits of fascicles. Fascicles are made up of subunits of fibers. Fibers are made up of subunits of myofibrils. And then in a myofibril, you have these repeating units of sarcomeres. And that is where a muscle contraction occurs at the microscopic level. And since in lab this week, we're going to be looking at muscle contractile force, that's a good place to start to learn how muscles contract. So breaking down muscle types. We are born with mixed muscle fibers, and muscles are plastic, which means they can adapt to change based on your use and demands of them. If you are a couch potato, your muscles are going to be primarily of one fiber type versus another. If you're very physical, an endurance athlete, your muscle fibers will have a higher percentage of certain fibers, so they can change over time. So red fibers are red because they're very pigmented. White fibers are white fibers because they're not pigmented. So the red fibers are your slow twitch fibers. Think of that for endurance exercise, okay? What happens is they're dark because they have myoglobin. Myoglobin is a muscle respiratory pigment. So if you've ever gone to the grocery store and picked up a package of, well, let's say beef, okay, and it's shrink wrapped in plastic, and you open it up, it's got a little pink fluid around it. That's not blood, you know. They take out the blood vessels. What you're looking at, that pink fluid, is muscle myoglobin. So it's perfectly fine, all right? And there's a really good blood supply to these fibers because you're really going to need a lot of oxygen and glucose. These are highly active muscle fibers. They have many mitochondria for making ATP. They have steady, but not big power. People who are endurance athletes aren't hulking masses, are they? Because that doesn't take you very far. People who run marathons, people who play tennis for hours on end, or soccer, or run up and down a basketball court for hours, they're lean, all right? Because of these muscle fiber types. They have steady power, but they're not going to be bench pressing 300 pounds, not likely, all right? They're for endurance activity. They fatigue slowly, thus you have endurance. And then you find it in the areas of your body where they're constantly being demanded upon, like your legs and postural muscles are working all the time, and your core muscles. Now compare that to, now I know this is not in your notes because I have it blocked out, but in the completed notes, this will be here. It's just the opposite of this. White fibers, I have the weightlifter there. They have less myoglobin, fewer capillaries. These are short burst power muscles. A lot of power, but they can't endure. Imagine, if you will, someone at the gym, at the bench press, and they should have spotters because people who bench press tend to do a lot of weight. And what happens is, yeah, they could bench press 200 pounds or something like that, right? Or do deadlifts or something like that. But the reason you have spotters is because you push those muscles because you're putting a lot of weight on, and you're only going to get a certain number of reps before the muscle runs out of ATP and then gives out. And then your spotter spots you. So they have fewer mitochondria. They're more likely to resort to anaerobic metabolism because they use up their glucose, they use up their oxygen, and then you build that lactic acid burn, and you know your muscles are going to fail pretty soon when you get that lactic acid burn. Short bursts of high power for short duration activity. They are not endurance muscles. They fatigue quickly. And so think about your arm muscles, and to some extent your leg muscles, but definitely your arm muscles. Okay. So since I want to get to muscle contraction for the quiz, I am going to go on, even if you haven't finished, but just put the opposite of all these things, okay? And then after class, if you want me to pull this up, I will. Okay, so some of this is going to be reviewed from chapter 4, which is nice. So you have a motor neuron. And since the rest of this chapter, we're now talking about skeletal muscles, we'll call this a somatic motor neuron. It synapses onto a muscle cell. All right, so here are the synaptic knobs right here, and there's the synapse, the space between that and the muscle cell. And so we know, because we just reviewed it, somatic motor neurons release the neurotransmitter, acetylcholine. The receptors, not shown very well here on that muscle cell, are nicotinic cholinergic. And I've covered this before, but let's see if you remember from me covering it back in chapter 4. Once the acetylcholine binds to its nicotinic cholinergic receptor on skeletal muscle cells, what ion channel opens up? Sodium. Okay, and that will cause an action potential in the muscle. Ultimately, it will contract. That's what you learn. Now we're going to, in this chapter, go over, there's a few more steps from when sodium channels are open, it comes flooding into the cell, and then the muscle contracts. There's a few more steps that happen before the muscle contracts, and that's what we're going to go over today. So I know it's a lot of review. God, how many times is she going to go over it? Until you got it memorized and you're set for pharmacology. That's how many times I'm going to go over it. So the neurotransmitter released is acetylcholine. It binds to its nicotinic. So if you get this wrong on the lab quiz, I'm going to kick you. All right. Cholinergic receptors. And opens up sodium channels. Sodium enters the muscle cell and causes an action potential, an EPSP. But I'm going to introduce some more steps before that muscle contracts. And part of that is this. The action potential causes this next step, which is new. I've not covered that with you before, new material. The action potential causes calcium release from a calcium warehouse in muscle cells called the sarcoplasmic reticulum. You might remember that from intro bio. So another ion is important in getting the muscles to contract. All right. Okay. So from here, I'm going to introduce the players that you've probably not been introduced before. And these are going to be the players that are involved in the muscle actually contracting. So actin is the purple thin filament here. And myosin is the big green blobby muscle filament. They're called muscle myofilaments. I just call them filaments. Actin, I put in purple to color code with this, is called the thin filament because it is thin compared to myosin. And here's how I remember this. If you stay active, you stay thin. Okay, whatever works. So actin is the thin filament and it has all these things right here, and you can't see them here. Have active sites. They have active sites which allow the myosin head to bind. I'll say heads because there's many. Okay, but there's some steps that have to happen. Troponin is the protein on actin. These are all found on actin, which binds to the calcium. All right, and I'll put in parentheses, and it's connected to the next protein, tropomyosin. Okay, so troponin and tropomyosin are connected, but only troponin binds with the calcium. So remember, the short T word binds with calcium, and the long T word is the one that normally covers the active sites. Okay. All right. So you're not going to have to memorize diagrams or anything. This is all vocab. All right. So what I'm going to show is first diagrammatic what's happening. Then I have a couple of GIFs, and then I have a link to a YouTube video, which you can look at. But we're going to talk about all the steps that happen after calcium is released. It's going to bind to troponin. Tropomyosin normally blocks these active sites that myosin wants to grab a hold of. So something has to happen to get tropomyosin off the active sites so that myosin can bind. And so that's what we're going to talk about. Okay, so diagrammatic representation. Here's our motor neuron, releases acetylcholine. The receptor is nicotinic. And then once the neurotransmitter binds, it opens up sodium channels. All right, you get an action potential form. So step 3, here's the action potential. It says the action potential goes down the T tubules, transverse tubules. And if it goes down here, that's the tubule. The green blobby thing is the sarcoplasmic reticulum. It's a calcium warehouse. So when that active potential hits here, calcium is released in a wave inside the muscle cell. And then what I had in the previous slide, it's going to bind to troponin on actin. All right. So this is a little GIF. Here's calcium binding to troponin. The purple line is tropomyosin, which is just lifted off to expose the little active sites. Once the active sites are exposed, myosin heads will grab and they will pull. And they will keep doing this as long as they have enough energy. All right. So here's the calcium binding to the blue thing is troponin. Since troponin is connected to the tropomyosin, the stringy thing, when it moves, it lifts tropomyosin off the active sites, then myosin can grab and it can pull. Okay. So imagine this tug of war. You might have played it as kids. Okay. Intramural sport. I don't know. But you've got the rope and you've got an opposing team, right? And so you have to grab the active sites on the rope to be able to pull, right? Well, in this case, that's what myosin wants to do, is grab a hold of actin and pull like it's a rope, all right? So in order to get those active sites exposed, calcium has to come in, bind to troponin, and then tropomyosin to lift off the active sites and you can grab and pull, all right? And I'm going to talk about the energetics of muscle contraction, because if you make one pull on the rope, do you make much progress? I was watching the SAG Awards, Screen Actors Guild Award last night. You want to watch it? You remember Squid Games, the actor got an award, right? No? You watch it? Squid Games? You didn't watch that? Where they tug of war over a pit, like 5 stories high, people fall to their deaths, right? Well, anyway, that made me think of the muscle and the tug of war. In order to make progress, you have to let go of the previous grip, go further down the rope, grab again, and pull again. Let go, grab, pull again. That's going to take energy for ATP. So we're going to talk about that. All right. So here is the energetics of the muscle contraction. All right. So just look at the GIF for a second. Right. I'm going to wait till it starts over, not this. Okay. So once the active sites are exposed, myosin head grab and pull, but they can't let go because they're out of energy now. And what breaks the connection is ATP, so they can let go of the rope. And now it's back to ADP, and they grab and they pull. So you need muscle ADP to grab a hold of active sites and pull. But once you've pulled, you can't let go until ATP comes in, breaks the cross bridge. Here it is, ATP, da, 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 boom. But now we're back to a lower energy, but we can still use that to grab and pull. So it's a repeated process of this. ADP, pull, grab, ATP, release, ADP, pull, grab, ATP, release. That's what it is. All right. So I've got a much longer, better YouTube video if you want to just, because a video can do so much more than me up there waving my hands and all that stuff. You see, you're like, oh, I get it. All right. So that's there. And the reason I have the people rowing is because they do the same thing. They put the oars in the water, they pull. And imagine they couldn't reach forward again unless they had ATP. That's how our skeletal muscles work. So I broke it down to something very simple. Oh, I have to do this. Let me back up. So let me put it here. You need ADP for, I'll just say myosin to grab and pull on actin. You need ATP to break, it's called a cross bridge, and if you look in the textbook, I think they talk about it, but when I say grab and pull, that pull they call the power stroke. That's a fancy word for it pulls on it. So I just say pull and grab and pull. And then ATP to break the cross bridge so you can reach further down actin and pull again and pull again. Now what happens is when myosin keeps grabbing on actin, the end plates of a sarcomere get pulled in, get pulled in, get pulled in, until the muscle is shortened, all it can shorten. And all those sarcomeres, thousands of them, as many as 10,000 back to back, that ends up in the whole muscle shortening. And when the muscle shortens, that's when it produces force, it changes shape, and it pulls on some object, all right? In lab, the handout, it says when the muscle shortens, it's called an isotonic contraction because the muscle gets smaller. All right, so look over that too in the lab handout. So what does this have to do with rigor mortis? I've seen rigor mortis. Has anyone ever seen rigor mortis? After usually mammalian body, but anything, insects are a little weird. About 12, 18 hours after death, the body stiffens. So, and you'll see it in nursing, unfortunately. Well, I've seen it in the animal world. What happens is the body stiffens because at the time of death, and you're going to see this, and it's going to be a little disconcerting for you, but you'll get better the more you see it. Usually people, when they die, sometimes it's nice and peaceful, and they're just like, then they're gone. But a lot of times, they'll go through brain death first, and the body goes through what's called agonal movements, where it'll twitch, or they might do little last-minute actions or something like that. They're gone out here, but their body is still going through the throes of death and cellular death. Don't freak out, okay? So that means there might be some muscle contractions going on at the moment of cell death. And when cells die, there's no more ATP being made. Dead things don't metabolize, right? And so all the muscle cells, all the actin and myosin cross bridges that were formed at the time of cell death, without ATP, you can't break the cross bridges. And the muscles stiffen up. Okay. So that's what rigor mortis is. No more ATP to break the cross bridges between the muscle filaments, and they stay there. About 24, 36 hours. I'm not a forensic person. The body relaxes. It's not because ATP magically got produced. It's because the body's beginning to decompose and actin and myosin are beginning to fall apart. All right. A not so gruesome way of looking at this. You might not be aware of it, but in the beef and other meat industry, but especially beef, because beef cattle are big. When they process animals for human consumption, they will actually hang carcasses in big refrigerated rooms, because they don't just process an animal and immediately cut it up and package it and send it out for consumption. It's not tenderized. Okay, they're waiting for about maybe 3 days to a week for all those muscle myofilaments to soften, for the meat to become tender. They're waiting for this to happen. All right, so not so gruesome. Maybe for some of you it would be gruesome just to think about that too. But yeah, that's what happens, is the muscle myofilaments break down. Okay, this gets into some lab vocab. So an isotonic contraction is the one on the right. When all those sarcomeres shorten, because myosin is grabbed and pulled on actin and it's pulled in the sarcomere as short as it's going to get, the muscle shortens. So it is a contraction where the muscle shortens. And another way to look at this is the muscle produces more force than the mass of the object being moved. Okay, let me explain this, because muscle stuff can get really heavy into the realm of arcs and vectors and physics and all that stuff. I don't teach it that way, because that's physics. But this woman's picking up a box. Maybe there's some things in the box. So that box has a certain mass. Maybe it's 20 pounds, maybe it's 30 pounds. I don't know. But her arm and leg muscles and core muscles, when they contract, it produces enough muscle contractile force that is greater than the mass of the object. The object moves. Hey, I just moved an object. My biceps muscles contracted a little bit. There's another way that muscles contract, and that's called isometric, where the muscles develop tension, but they don't shorten, usually because the mass of the object you're trying to move is too great. Okay? I guarantee you my muscles are generating tension, but there's no way in hell I'm going to be able to lift this podium. Okay? Its mass is too great for me. So that's an isotonic contraction. Isometric exercises. My muscles have tension, but I'm moving either way. Okay, that's why it's called isometric exercises. So the muscle generates tension, but doesn't shorten. Okay, the reason I've got the guy playing like he's lifting a boulder is there's no way in hell he's going to move that boulder, but his muscles are generating tension, but there's no way he has the contractile force to move that. All right. So those are two basic ways that muscles contract. And I'm going to talk about other ways, other things that affect muscle contractile force. Because in lab this week, you're going to be squeezing a muscle dynamometer. It's bigger than this. And you're going to actually measure the force that's produced by your grip strength. All right. So it's relevant to lab this week. So we did good. So we basically got through this, which is what the first page of the lab handout talks about. So when I ask questions on that and players involved in sliding filament theory, you'll have been exposed to it. Okay. Okay, I feel good about that. I wanted to do that. So this is just more muscle stuff. Vocabulary. This chapter is all vocab. The only sequence that you're going to need to know is what I just covered. A motor neuron releases acetylcholine into the synapse of the muscle cell. It binds to its nicotinic cholinergic receptor. This is written down. Sodium channels open. Active potential forms in the muscle cell. Action potential goes down the T tubule. Causes calcium release by the sarcoplasmic reticulum. And on the slide where I had actin and myosin, tropomyosin normally blocks the active sites on actin. But when calcium binds to troponin, that lifts up tropomyosin off those active sites. Myosin grabs and pulls. And then there's ATP, break, cross bridge, ADP, grab, pull. That's it. That's the sliding filament. I could ask on a future exam, and I probably will. I will. What's the sequence of events that happens from the release of acetylcholine from a motor neuron to a muscle cell contracting? And you'd have to put them all in the right order. Okay. So, motor unit is the motor neuron and all the muscle cells, and muscle cells are fibers, remember. It innervates, and that's a fancy word of it connects to. So we've got an example of a motor nerve right here, and it's connecting to 1, 2, 3 muscle fibers. That's a motor unit. All right. Now, there's a different arrangement of motor units in your body. I was watching this cool thing on PBS, and probably none of you ever saw it, but it was so amazing. My husband was screaming, "Tam, get in here, you're gonna love this." It was a guy who lost his lower limbs to frostbite, bad frostbite. Anyway, so they cut his legs off below the knee. And he then said, well, I'm not just going to feel sorry for myself. He went on to get a PhD in neurophysiology at Harvard. And he's doing all this cool research about reconnecting the nerves that remain at his stump, basically, to muscle fibers, or that's actually synthetic neural units in a prosthetic leg that has electronics. All right. So that, by just his thoughts alone, his motor command to the remaining nerves he has, and it synapses onto this synthetic leg, he could make the leg do what he wanted. It's not just this static thing. It was cool. It just blew my mind. But anyway, I'm excited about this stuff. I think it's really amazing. But in your core muscles that support you and that produce a lot of strength, leg, gluteus muscles, all of those, you can have as many as 150 muscle fibers innervated by one motor neuron. And that arrangement of a motor neuron, one neuron, many muscle fibers, is going to produce a lot of force. Okay, you can do all these fun things, you know, without falling off the stage. In your fingers, okay, if you're going to test your tape strip, they do not produce as much force, but you can do such delicate work. All right, not much force, but great precision. Whereas the core muscles, a lot of force, but not so much precision. So that's an arrangement of muscle motor units. So power versus precision. Okay, so let's look at it. Muscle precision is one motor neuron connected to few muscle fibers. Not much power, but you have precision. It's a trade-off. And that's it. It's just a trade-off that our body has. If you need a lot of power, you'll have one motor neuron connected to many muscle fibers. Remember I said there can be as many as 150 muscle fibers. You have power, but less precision. Except for patients who have lost their upper limbs or were born without them, and then they can do all these amazing things with their feet, right? But that takes a lifetime of training. So when I'm looking at the blue motor unit and the red motor unit, okay? So here's a motor unit, and then here's a motor unit. Obviously, the blue has 2 muscle fibers, and the red has 1, 2, 3, 4, 5, 6, 7, 8. 8. The large one's going to provide more power but less precision. The small one has less power but more precision. And they're arranged that way in our body because of the uses demanded upon them. Okay, so it's just a vocabulary, just concept. Now, what affects muscle contractile force? In lab, we're going to test this. You're going to experience, after we do your grip strength versus your dominant versus your non-dominant hand, because over a lifetime, if you have a preferential hand that you do most of your stuff with, that's going to have more power, possibly also more precision in the fingertips because you use it more often. Okay, we're going to look at that concept. We're going to look at the strength of our different fingers, which is the power versus precision rule. And then lastly, you're going to look at muscle fatigue. Yeah. So you're going to grip this sensor for 100 seconds. You're going to find out exactly how long 100 seconds is. And you can, well, your partner who's running a computer will watch as time goes on, muscle contractile force goes down, 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 because your muscles are fatiguing and producing less force. Okay, you're using up oxygen. So here are the things that affect muscle contractile force. How many fibers is the neuron attached to? Okay, if more fibers respond, you have greater force produced. Okay, this gets back to the motor unit arrangement. If you're motor unit X and you're connected to 5 fibers, you're not going to produce as much force. If you're motor unit Y and you're connected to 7 fibers, you can potentially produce more force. And the opposite goes true. If there's fewer fibers, you produce less force. So this is not hard stuff. Okay, very common sense. The other thing is, how strong is the stimulus? Here is what the stimulus strength is. How much acetylcholine is in the synapse? That's it. Okay, so I'll say strength, and I'll put how much ACH is in the synapse. That determines. Remember how we've covered all of this? The more neurotransmitters in the synapse, the more of a cell response you get, the less neurotransmitter in the synapse, the less of a response you get. I covered that. Okay, I covered that in chapter 4. If the stimulus is strong, a lot of acetylcholine, you get greater force produced. And if the stimulus is weak, there's only a little bit of acetylcholine, you get less force. What is this called? Because it's not on the PowerPoint. I gave you 2 ways that you can have an EPSP, an excitatory postsynaptic potential. And I said, if there's a lot of neurotransmitter versus a little. I gave you the example of a woman who's breastfeeding a child has oxytocin release, a little bit. And so there's milk let down, but the uterus will contract just a little bit. So that little bit of oxytocin. Childbirth, you have oxytocin release, a lot of oxytocin. You get big uterine contractions, big response from muscles, okay? What is that concept called? Graded potential. Okay, a graded potential. In the lab this week, you're going to have that endurance exercise where you're gripping that muscle dynamometer. And it's 100 seconds long. And from time 0 to time 80 seconds, the force is going to go down. You're going to see it on the computer screen. Then what you're going to do is your partner to the person who's squeezing the thing, is going to become a cheerleader. Come on, keep going, squeeze, 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 squeeze. Mentally, you know what that's going to do? It's going to cause more acetylcholine release. And we'll see it. We'll do it as a class. We'll get the class data. From 80 to 100 seconds, you'll see it start to go back up again. Because psychologically, you're causing more acetylcholine release in your partner. And it's kind of cool to watch. All right. So yes, we are looking at these things in lab this week. Now, oh, it was there. Dang it. I should have left it off, but now, if there is a big need for a muscle contractile response, and you need a lot of force, that is more than one motor neuron can supply with all the muscle fibers it's attached to, what you'll get is this. You'll get multiple motor units respond. So you get motor unit X plus Y, maybe plus Z. And that's called recruitment. Because you're recruiting more than one motor neuron. Everything I've got right here is for one motor unit. Okay, but what happens if that's not enough force? You recruit other motor neurons, and that will help it. And they can do that. They can join forces together. Think of it this way. If I ask someone to come up on stage and lift me, I'm not a tiny girl. One person might be able to do it, but it'd go easier with recruitment of multiple people, motor neurons, and then you can get the job done. All right, that's recruit. We're doing good. Everyone, the pace is okay? All right. Turn the page. Okay. So we covered how many muscle fibers respond, that affects force, how strong the stimulus is, how much acetylcholine is in the synapse, and then frequency. Is it a frequent stimulation or acetylcholine binding to that receptor? So if it's, so what I've got here is, here's a stimulus. This is a muscle contractile, or I'll call it a muscle AP. The muscle contracts, and then you get another stimulus, and it contracts again, and another stimulus, it contracts again. That's like a stimulus every now and then, and you get a response. A single stimulus and a single contraction is called a muscle twitch. Okay, this is a twitch. This is a twitch, and so on and so forth. Okay, now if you start to up the frequency, like let's think about this. You've got one motor unit, a motor neuron, all the muscle fibers it's attached to, and it gets one stimulus. The neuron releases acetylcholine, and it binds to a receptor, and you get muscle contraction. Let's say it's an infrequent stimulus. It's like stimulus, stimulus, stimulus. That's very infrequent stimulation. The muscle force that's produced is very low. Like a single twitch is the lowest frequency. It's one stimulus, one contractile response. If you up the game and you have repeated low frequency stimuli, so it's like this, stimulus, 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 stimulus. That's more frequent. So here's the stimulus. Here is the response. Another stimulus. Here's the response. And notice what's happening. Keep this in mind. Because in nursing, you might see patients with neuromuscular issues, and there are simple tests that can be done, like an EMG, a muscle electromyogram. And what you want to see in normal muscle is this pattern. It's called muscle treppe. When I think of muscle treppe, I think of muscle stepping up. Stepping up or warming up. I have it right there, so I don't need to warm up. When you contract your skeletal muscles, when acetylcholine stimulates it, the muscle contracts, and it has time to go back down to baseline because it's not that high frequency. It has time to rest before it's stimulated again. Next time it contracts, it's going to produce a little more force. It goes back to baseline, it rests, stimulated again. Notice how it goes up. Now, eventually, not really shown well on this, is that muscle group will reach its plateau where just because of a number of muscle fibers responding, that's the maximum force this muscle group can produce. That's called treppe. This is normal. If you see a patient where after repeated low frequency stimulation with external sensors, they can do this. If it goes down instead of up and then plateauing out, that's abnormal. Okay, so you do this when you have people who have muscular problems to try and rule out what's going on with their body. So the big thing is with every contraction, it goes back down to baseline, it gets to rest before it's stimulated again. You can push your muscles harder than that with the next category, but the muscle starts fatiguing. This is like your endurance long distance muscles can do this for a good amount of time before they give out because it's just low frequency. It's not too much of a demand. Your muscles can do that. Now, if you push your muscles a little bit harder, more frequent, okay, your muscles can respond, but look, it responds, but it doesn't have time to go to baseline before it's stimulated again. Doesn't have time to rest, stimulated again. And it will peak out as well. And that's called muscle summation. And I covered that in the chapter on neurotransmitters. You might not remember, but I did. The frequency with which a neurotransmitter is released and binds to its receptor, if it's a low frequency, you get a low cell response. If it's a higher frequency, you get a higher response. In this case, more muscle contractile force, but your muscle's going to fatigue at some point because it's not getting to rest. Now, the highest that you can push your muscles is muscle tetanus. Oops. And we talked about tetanus when we were talking about toxins, tetanus toxins. But you can push your muscles this way. This is like those white fibers that produce short bursts of high power, but they can't keep it up. They're going to give out. That's what we're talking about. It's the highest frequency, right? The muscles shoot up to the highest power they can produce. But guess what? You're going to crash. You need a spotter or the bar is going to fall on your chest. You're going to die. Well, no. But you can put out that huge muscle power in muscle tetany with the highest frequency of stimulation, but the muscle's going to give out. It's just going to fail on you. So our muscles can do all of that. Okay. But so that'd be the graph representation of that normal muscle activity. How we doing? Great place to stop for today. So I covered what I definitely knew you needed to know for the quiz. So don't forget that. I'll see some of you in lab today.